Great.
Right.
Thank you. So we'll give, probably a couple of minutes, because Zoom meeting normally, people, not, joining, jump because of another meeting and so on. Actually, I gave a talk yesterday, 11 p.m. to midnight-ish, in some another, international one. And then there are many, keynote or even the preliminary one, including, Jim Collins and Dave Baker. I think you also used to be, you still in Seattle, right?
I used to. I just moved to New York.
Oh, so New York. So okay, so today in the case
just
of New York,
just like 6 weeks ago. Just 6 weeks ago, yeah.
Oh my goodness.
Where in New York are you?
I'm living in Bronxville, but the headquarters is downtown Manhattan. So I'm a New York commuter now. So
okay.
Oh my God.
3 days a week I go to the office. The other days I sit here at home. But
oh my God, I envy you both. Because oh my God, I love New York. I've been there many times. For musical and for musical, of course, and whenever my mother or my mother-in-law visit, Boston at the time, I can always go to New York. Actually, when my baby was born, my mother and my mother-in-law actually visit, to support us, and then after time, and then they want to go back to Korea, and then we always go to New York first. Because transferring for the older people, without any, language kind of, ability, in English, it's kind of, very challenging. So I normally kind of drive there, and then just kind of see around, and then after that, we basically stay one night and then go back to Korea, and then I go back to Boston at the time.
And
wow, Boston, New York.
Interesting. You're out in Long Island, I guess, or you live
I'm at the NYIT campus out on Long Island, but I actually live in the Upper West Side. My wife, so I do the reverse commute. Yeah, my wife is she's an elementary school teacher in the Bronx.
Oh my God!
So commute-wise, the Upper West Side is sort of an hour for each of us. So she takes the D train up, and actually it gets the B and the D. And then I take the LIRR out to Long Island.
So that means you're a neighbor, real neighbor.
Kind of. Bronxville is that's like in the suburbs. So
okay.
Well, actually, that would be an hour to get down to where you're at. Yeah, out to Long Island is about 2 hours, I think, by the time.
Yeah. You have to cross to get to Long Island from where you are. Actually, I have a colleague who's actually up in Bronxville, and he commutes out here, but he goes over really? Yeah, he takes the Broad's Neck Bridge.
Ah, okay. Yeah,
right, and the Whitestone Bridge. So one of those.
Oh my God! I
learned
now,
my geography.
Now so surprised.
But there's no train or anything between.
There's no train. Yeah, I was thinking on the train because
right. But I think
the train that comes by our place goes to Grand Central pretty much.
Right.
Oh my God! So I was shocked right now because I have so much interesting kind of coincidence during this seminar series, and actually one of the seminar actually happened September 23rd, and then that was my wife's birthday, and I just joined the after talk Zoom meeting, and then I have the one person kind of joining that meeting, and then I found out his kid birthday actually the same day we met for the first time. I didn't know about him before. And then we just met for the first time, and then he talking about today's his kid birthday. What's the probability? Nathan, you are the math genius. What's the probability and having two persons in the same Zoom room, same day? Oh my goodness! You are now talking about you are the New York. Okay. Anyways, we probably need to start. Oh my goodness. Okay, so we'll start. So good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. Hello everyone. Welcome and thank you for joining us, the 7th seminar and Health-Onegevity Day. As you know, this week is special with Nobel Prize announcement. In fact, I did wake up 3:30 in the morning yesterday and watched the live announcement 4:45 a.m. my time. Nobel Prize in Chemistry went to Drs. Benjamin List and Dave Macmillan, who developed, quote, a new and ingenious tool for molecular building, organocatalysis. Its use include research into new pharmaceutical, and it has also helped make chemistry greener, unquote. Also, Nobel Prize in Physiology or Medicine was awarded jointly to Drs. Dave Julius and Ardem Patapusan. For your information, Ardan was born in Beirut, Lebanon. They discovered key mechanisms of how people sense heat, cold, touch, and their own bodily movements. So I would say warm and soft are what we all feel when we are born from a baby and mom. Cold and hard are what we all feel when we let our beloved ones leave this world to heaven. We lost many people because of COVID, and I'm basically thinking about this kind of thing today. Asymmetric organocatalysis, the topic of Nobel Prize in Chemistry this year, is critical for safe and efficient drug development. I know this very well because my bachelor thesis topic is about asymmetric synthesis using enzymes, while this year's prize is the third type of catalyst, small organic molecules, a little bit different. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Nathan Price, CEO of Health-Onegevity, a division of Thorne Health Tech, and a professor at the Institute for Systems Biology. He was selected by the National Academy of Medicine as one of the 10 emerging leaders in health and medicine. He was also the chair of the NIH study section on modeling and analysis of biological system, MAPS. And what else do you want to know? Professor Nathan Price, not Nobel Prize. The floor is all yours.
Great. Thank you so much. And it's a pleasure to be with everyone here. And I want to thank Taesiak for putting together a great series and for the chance to talk to particularly some of the young people that are getting interested in synthetic biology. And I'll share just a little bit of kind of my, a little bit about what I've done, a little bit about my journey. But I think it's just, couldn't be a more exciting time for so many of you that are engaged in this field. And, as we are hopefully coming out of COVID here fairly soon, we've had this, big, huge impetus into the healthcare system that has led to a number of changes, I think. And one is that there's been a much deepened realization that we can change the healthcare system faster than we thought. If you look at an area like telemedicine, right, we probably accelerated 10 years of telemedicine in about 4 months because of necessity. Vaccine creation went from this, long multi-year process to getting done in a year. Now there's reasons for it in terms of the resources and barrier dropping and various things like that. But the value or the ability to see what we can achieve with real innovation and motivation in healthcare has been pretty astounding. I think the other big change that we've seen from this is that there's a bigger appreciation for the need in healthcare to focus proactively on wellness and prevention. And one of the reasons that I talk about that, even related to an infectious disease, is of course, if you look at the comorbidities of people who die from this disease, and it can hit anyone. So I don't want to be overly prescriptive about this, but there's many health conditions that raise your odds of having a death or a serious negative outcome from something like COVID. And so the reason I bring that up is, so just to give a little bit of my background for those that don't know me, like my early days were really spent doing metabolic modeling, geared towards optimizing different kinds of maybe bacteria for production of products and things like that. That's a pretty common in synthetic biology. I did a lot of systems medicine work for Alzheimer's disease, certain conditions of pregnancy, cancers. But for the last about 7 to 8 years, a lot of my effort has gone into this new area that we call scientific wellness. And the reason for that has been that I've become really convinced that the way that we go about dealing with chronic disease is often not put quite in the right place from my perspective. There's a lot of reasons that we do what we do, but this whole notion that most chronic disease can be dealt with a single target drug administered late in that process, I think is not likely to be highly successful. I don't think it's our best strategy. And so that has led me to really be interested, and this was mostly done in partnership with Lee Hood at ISB, who's been just the great, first mentor for me, and then partner later on all of these kinds of things. And basically, the view is to try to understand what happens in the years and even decades in some cases before the onset of serious chronic disease that you can focus on and optimize health and readiness so that you are more resilient to what comes later. Lee and I became super interested in that. We launched a project we called the Pioneer 100, which we took as a wellness project for, well, just like it sounds, about 100 individuals. It ended up being 107. And basically what we did is take them through a program where we were doing whole genome sequencing, and then multiple times per year, we'd measure proteomes and metabolomes in a whole bunch of clinical labs, microbiomes, and had them work with health coaches. And eventually, actually, we even spun a company out off of this so that we went from 100 people to about 5,000. And we did that for about 4 years. And what was remarkable from that, from my standpoint, was the amount of information and insight that you could glean by looking at what happens in all of these systems in individuals as they're going through kind of the regular course of their life. Now, we talk about it as a wellness cohort, but it's not, right? Not everyone is well, right? Some people get sick, some people develop disease. And so just to give the very briefest of this, you can read all, we have tons of published papers out of this cohort, but there are some really fascinating insights. So for example, there are certain markers like CCAM5, which is a known marker of metastasis, but we saw it elevated up to 2 years in advance of diagnosis in individuals coming through this program. So there's signs that seem to show up earlier on. The ability to change certain markers through lifestyle intervention, LDL cholesterol being a really interesting one. And it turns out that you can predict LDL cholesterol pretty well from genetics. There's a big genetic component. And so what we saw was that if people had elevated cholesterol where they'd be treated the same in today's healthcare system, if there was a gap, in other words, there was a delta between the genetic prediction of LDL and where they were able to very successfully lower LDL cholesterol by lifestyle intervention. But if their genetics predicted them high and they were also high, we saw no ability for people to actually lower it by lifestyle intervention. And so it immediately, this kind of data starts to give you clues and insights into the kinds of strategies that might work for a certain person, whether or not that would be drugs, whether or not it would be, some sort of natural products, whether it would be lifestyle interventions or whatever it was. So we found that really fascinating. The other thing we could do with this kind of multi-omic data was to use it as and ability to understand new data types like the microbiome. So we published a paper in Nature Biotechnology in 2019 that showed that you could predict the diversity of a person's microbiome from a pretty small set of metabolites out of the blood. And in fact, that led us to really start focusing a lot on how do you interpret the health of the microbiome? And even though the microbiome species are so diverse, there's a much more finite set of chemicals that appear in your blood that are made by the microbiome, both for good and for bad, that have known health effects that are positive. And positive like butyrate has a bunch of positive effects, or negative like TMA that gets turned into TMAO by your liver and is a cardiovascular risk function and so forth. We also delved in on aging, and we're able to identify in the microbiome that a person's microbiome, if you stay healthy throughout your life, it actually becomes more and more unique to yourself, i.e. looks less like anyone else's microbiome. But even as that diversity is increasing, that uniqueness is increasing, you in fact still have these commonalities in terms of the metabolic transformations that are catalyzed by the microbiome associated with health. We published that in Nature Metabolism this year, and that was received an award from the National Academy of Medicine, Health-Onegevity Catalyst Award, so they help support that. Anyway, there's a whole host of these kinds of ideas and efforts, and that's just a teeny bit of what's there. But the point I want to make here is that, as you think about your career and all the kind of things that you're going to do in synthetic biology, which is such an exciting and wonderful field, is that I would urge you to, as you're thinking about that, not only consider all the different disease cases where that can be applied, but also to think some about upstream, what kind of things could be done, in a microbiome, in whatever way that you can, that is aimed at those earliest processes of maintaining, optimizing health, and understanding those earliest transitions. Because it's my view is that systems approaches that are aimed earlier rather than later, which what we call scientific wellness, is going to be an incredibly important element for how we deal with chronic disease in a way that I hope will be a lot better than what we do today. And I guess I'll leave it there, and thank you for everyone's time, and thank you, Tae-Seok, for the invitation. I look forward to hearing what I'm sure will be a fascinating talk from Navin.
Thanks so much. My goodness, for your insightful, timely, important talk. Personally, I hope, of course, everyone, COVID, chronic disease will be gone and eliminated. And also personally, as one of the chronic disease patients for many years, and also one of the victims of the misdiagnosis, and also biomedical, also microbiome engineer, I really fascinated by what you just said. And thank you so much for your wonderful talk. So okay, so now of course the main speaker of today with a little bit longer introduction. Navin Pokala is an assistant professor in the Department of Biological and Chemical Science at the New York Institute of Technology, a primarily undergraduate institution on Long Island and New York City. And also I just found both Nathan and Navin basically, right now, in New York. I mean, what a, fate. I mean, I just found out, and then Nathan moved to, to New York. So of course, I mean, envy you, both of you, and why not? So he did his undergraduate work at MIT with Paul Masudaira and mapped the calcium and actin binding structures of fibrin, a cytoskeletal protein. For his PhD with Tracy Handel at the University of California, Berkeley, he developed fast and accurate continuum electrostatics and solvation models for computational protein design. For his postdoc with Corey Bachman at the Rockefeller University in New York City, he switched the field and learned how to use C. elegans as a model system for neuroscience. He developed software for automated tracking of animal behavior and pioneered the use of intersectional method for transgene expression in C. elegans. He will talk to us today about his lab's work developing histamine as an orthogonal synthetic neurotransmission system for C. elegans. I'm particularly interested in this talk because I have been working on neurochemicals and gut relationships, so-called microbiota gut-brain axis. And common interests, of course, among both speakers today and me made me call today as Health-Onegevity Day. Also, both speakers have the same initial, NP. So what a fate or fantastic match. Okay, Navin, take it away.
Let me just share my screen here. All righty. I want to thank Taesuk for inviting me to present our work today. And so I'm going to talk to you about our work we've been doing on trying to develop a synthetic neurotransmission system. So before I continue, I just want to highlight some of the people who have been involved in the work. I started this work when I was a postdoc with Corey Bargman at the Rockefeller. And so these are some of my colleagues who contributed. And then since starting at New York Tech, which is our undergraduate institution, I've had like dozens of undergrads involved in the work, doing cloning or making transgenic lines. And so these are some of the students that have been involved. And so as synthetic biologists, I think we all have a deep appreciation for this cryptic last comment by Richard Feynman on his blackboard just before he passed away. What I cannot create, I do not understand. And so the idea is that if we understand something, we should be able to use that understanding and build stuff, hopefully cool and useful stuff, okay? And so I'm a neuroscientist, and so what does it mean to like create the brain, right? I mean, the brain is already there. But I think of creating the brain or engineering the brain is really trying to, in terms of, is to try to design therapies that are useful a priori. So if you know something about a person's genetic history and their personal history, in principle, right, you should be able to design like the right set of therapies to help them out. But unfortunately, most of the therapies that are in use were all the result of accidental discoveries from the 20th century. And then, there's been some, improvements, right? In terms of less reduced side effects and stuff like that. But we really don't have a really good understanding of how the brain works at all. And so what we would like to do is be able to understand how we go from molecules, and we understand how drugs interact with the proteins and receptors and nervous systems, to go from molecules to cells. And again, we understand the cell biology of neurotransmission and stuff really well. And then understand how these cellular networks lead to behaviors. And of course, people have been studying psychology as a field for about 150 years. And then, of course, before that even. But actually linking all these up, I think, is the key for having a deep understanding that could then let us actually engineer therapies, okay? And so I'm really inspired by the tremendous progress that's happened in protein and enzyme engineering. And so this is one of my favorite classic papers from Carter and Wells from back in 1988. And what they did is that they took acetylysin and they mutated all the catalytic triad residues, including the catalytic serine. And so using a brand new technology of site-directed mutagenesis. And so they were able to then better understand the catalytic mechanism of cetylysin. And then 20 years later, David Baker and his colleagues were able to, basically take an enzyme reaction that did not exist in nature and then use a computer program and then design an enzyme that would catalyze that reaction. And so that's the dream. We'd like to be able to do something like that, take, our basic understanding and then design something useful, hopefully for patients. And so there's some parallels, I think. With enzyme design, a lot of progress was made because the structures of lots of proteins. In neuroscience, now there's a lot more known about, we're developing knowledge about connectomics and how cells are connected, and the wiring diagrams, and then also the mechanics of how they communicate with each other. One major step for enzyme design was being able to design and generate any mutation or sequence at will using site-directed mutagenesis and gene synthesis. With neuroscience, we have these technologies like optogenetics, chemical genetics, and now genome editing that now let us change and change any neuron or activate any neuron or inhibit any neuron at will, at least in theory. Okay. Then there are also importance of having assays. And so you have enzyme assays, protein stability assays, and understanding how these effects of mutations affect the function was important. And then likewise in neuroscience, you were able to record neural activity and then analyze the behavior of the animals. But then I think another important aspect of enzyme design, and that was important for success, is understanding design fails, because there are a lot of design fails. There's a lot of design fails in just designing a protein that folded up, even if it didn't do anything. And so there's, so I'm going to tell you some today about our efforts in chemical genetics, which I think is analogous to making site-directed mutants, back in the 80s. And then a little bit about some of our design fails and what we've learned from that. And so I don't think it's going to take, I don't think 20 years from now, we're going to be able to say, okay, take a patient who has some problems and then have the exact perfect drug for them or the perfect therapy for them. But hopefully we'll be better than where we are now. And then maybe, years from now, that'll be there. So a lot of small steps, and that's the start. And so the model system that I'm using for this is C. elegans, which is a small nematode. It's only about 1 millimeter long when fully grown. It grows from a single egg to an adult with 959 cells in 3 days. They eat bacteria. They're very cheap and easy to grow, which is really important, especially at an undergraduate institution where you're working with undergrads. It's very easy to make transgenic animals. There are a number of behaviors that it does. Some of them are very reflexive. Others are more complex, like exploration, learning, and even social behavior. And it manages to do all this with a relatively simple nervous system. It has only 302 neurons with only 7,000 synapses and gap junctions, whereas we have we have trillions of connections and 86 billion neurons, and so this is a way simpler model. And so if we can understand how to do stuff with this in a predictable way, hopefully we can apply those lessons to bigger critters like us. And so this is the wiring of the C. elegans brain. And so there's an overall flow from sensory neuron to interneurons to motor neurons, but it still is rather complicated. And so even though this is a highly simplified nervous system, it's still way complicated. And this wiring has been known since 1986. and we're only barely now beginning to understand how this might work. And just in comparison, a cubic millimeter of cortex in the human brain has 10 to the 5th cells. And so this is way simpler than a cubic millimeter of a human brain. And so it's a big challenge. And so understanding, if we don't understand this, there will be no hope of understanding or engineering that. Okay. And so one of the most powerful tools that we have is that we have promoters that allow us to drive the expression of transgenes in single cells. And that's been incredibly powerful. Okay, and so today I'm going to tell you about our efforts to use histamine activated receptors as chemical genetic tools for C. elegans. And then, and some of that's been published. And then I'm going to tell you about some newer work on trying to use histamine as a synthetic neurotransmission system in C. elegans. And so what we showed a few years ago was that we could take this HisCL gene from Drosophila, and which is a histamine-gated chloride channel. And then if we express this in worm neurons and then add histamine, the neuron that it's expressed in becomes hyperpolarized. And so that basically means that it becomes inhibited and is less able to then signal, okay? And so this is an electrical recording. And so this is different from the histamine receptors that we use, which are G protein coupled receptors. Insects use histamine-gated ion channels, okay, like this one. And this is pretty neat because worms actually don't use histamine or respond to histamine. And so, and for me, this is a really, seemed like a really great opportunity because we can now, just by expressing this in a worm neuron, silence it at will using histamine delivered, in the media. And histamine is really, really inexpensive. It's also friendly and compatible with optogenetics and calcium imaging. And like I said before, it's cheap. And so you could just pour liters of agar plates and do genetic screens using this. And so it's become a very well-established and popular tool in the worm neuroscience field, okay? And so one of the behaviors that we study just to sort of prototype this is the touch response. And so if an animal's crawling around and you touch it on the head, it basically backs up and turns around and runs away. And so this is part of the circuitry that's involved. And so here's the touch sensory neuron, which then sends outputs of some of these cells. And so what we did is that we expressed this HisCL channel in these cells and then asked what is the effect when we silence these cells individually on this response. Okay, and so when we silence this AVA cell, what happens is that the worms, when they get touched, they pause and then they make a turn, but they fail to reverse. And so what that tells us is that this cell is required for triggering reversals. It's not a motor neuron, but it signals to motor neurons. There is another cell called AIB. When we silence that, the worms are able to reverse, but now they lose the ability to turn in response to the touch. And again, this is not a motor neuron, but it signals to motor neurons. And so this is part of the circuitry that we've managed to better understand using this. Okay. Another experiment we did had to do with trying to understand a more complex behavior, which is chemotaxis, which involves animals looking for food and trying to find food odors. And so here, this is work done in collaboration with my colleague, Andrew Gordis. And what we did is that we expressed the calcium indicator protein GCAMP in these three neurons, AVA, AIB, and RIM, which are interneurons. And so AIB gets input from a sensory neuron called AWC. In AWC, its responses are regulated by odor. And then what happens is that when you deliver odor, these three cells actually become inhibited by odor, okay? But you'll notice here that this response is rather variable. And so only around half of the time does a given cell get silenced by the odor. The other half of the time, there might be a delayed response or no response at all. And so you get some variability there, okay? And so what Andrew noticed was that, if you have these three cells, these are the different possible network states that you could have. But what he noticed is that most of these states are never observed. These are the only states that he would ever see in a normal situation. And so what we did is that we synthetically created all of these states using his CL. So we can create these artificial brain states when we do that, okay? And then so here, and again, here's the normal situation where you get around half the time you get an odor response. And then when we express his CL in RIM becomes silenced. And then we ask what's the odor response in the remaining two cells. And what we found is that now the remaining two cells, this AVA and AIB, they respond with much greater fidelity to the odor. So basically 80 to 90% response instead of about half, 40 to 60% response. And so what it seems to be happening is that this RIM interneuron, its normal job is to somehow increase variability of odor responses. And we could see that by if we silence it, then the variability goes down. And it turns out that this might be a way of, basically deliberately increasing variability might be an advantage for actually behaving in a noisy world where you actually might want to have a variable response to deal with this crazy world we live in. Okay. Another thing we've done to follow up on is, where his CL silences neurons. We engineered a cation variant of this channel by making some mutations here. And when we do that, and we express this in the same AVA neuron, that if you silence it, the animals can no longer go backwards. Here, if we express this cation variant, and you put the animals on histamine, now they back up all the time. And so we've created a cation version. So now we can activate these neurons using histamine. We've also taken the histamine receptors from mammals that are not chloride, that are not ion channels, but rather are G protein coupled receptors. And each of the mammalian receptors acts through a specific G alpha subunit. And so what we've shown is that we can express these in C. elegans neurons, and then they act through the different G protein signaling pathways. Okay. So now we have these tools for now regulating the activity of a single neuron, either inhibiting it with the chloride channel, activating it with the cation channel, or triggering these intracellular pathways using the G protein coupled receptors. But this is part of the worm's wiring diagram. And what you can see is that if you affect the activity of this neuron, you're affecting all of the connections that it makes. And what we would like to do for engineering purposes is to just affect a single connection, okay? Or maybe create new connections and perhaps new functions. And truth be told, we're not the only ones that have been dabbling in this area. A few years ago, Bill Schaefer's lab, they showed that you could express gap junction proteins and C. elegans neurons and create an ectopic gap junction, which is excitatory. And so then they're electrically coupled. So it's non-synaptic. And then more recently, Daniel Colon-Ramos's group showed that you could take a peptide from Hydra, and that has a receptor that is a cation channel, and showed that you could release the peptide from one cell, and then another cell that has a cation channel will then respond to that. And so these, I think, will be valuable additions to this toolbox and complementary to some of the work that I'm going to discuss now with histamine. And so as a neuroscientist who has this wiring diagram, something we'd like to be able to know is how important are these different connections? And so this is this ASH sensory neuron, which responds to repellents. So when this neuron gets activated, that basically triggers the animals to get away. Okay, and so it has a number of connections to many different cells, including this AVA cell that I mentioned before that's required for reversals, but it has a bunch of connections to a lot of other cells. And what we would like to know is how important are these different connections? We can do whole brain imaging of every neuron in the C. elegans brain at single cell level, but we don't know if activity correlates with anatomical connection strength. There doesn't seem to be much of a correlation there. And then finally, we want to know how important is connection strength? Are stronger connections more important? Or is it not the connection strength, but rather the topology and the geometry of the connections? And so the way we can address how important a given connection is to try to address these questions is to cut an individual connection and do that. Okay. And so before I'm going to tell you about the experimental aspect, I want to tell you a little bit about some modeling I've done. And so what I did is that we created a sort of a toy neural network which has worm-like behavior. And so when you give it repellents, it turns around. If you give it attractants, it goes towards the attractants. And if it loses the attractant, it turns around to try to reacquire the attractant. And so we evolved a number of neural networks basically by adjusting the connection strengths, the polarities, and then trained it to mimic real behaviors. And then using this, we can then do virtual experiments to better guide real experiments with real animals. And so these are real animals in this panel using their real brains, chemotaxing toward a food odor here, okay? And so the way they do that is that when they're going towards food, if the odor, they have fewer turns, but then if they lose the odor, they will then increase their turning frequency. And so our simulated network here does a similar sort of thing. If it seems to be going away from the odor, it turns around to try to reacquire. And here we're able to do virtual whole brain imaging as it's doing the behavior. Okay, and because it's a simulated system, we can do any experiment we want. And so, so something I did is that I said, well, let's see what happens if we cut each individual connection. All right, and does the connection strengths, how do those matter? Okay, and what I found is that when we cut individual connections and then look to see how much time it takes to chemotax, there's no relationship at all. You can cut some very strong connections and have no effect at all. You can cut some very weak connections and then it basically just doesn't work. And so yeah, this is Yeah, so this basically sort of a hint about some of the behavioral complexities we might have to be dealing with in real animals. Okay, and so what we want to do is then work with these simulated animals and then try to better guide the experimental side of things. But now I want to tell you about how we can even do this cutting of an individual connection in real animals. Okay, and so because histamine is something that is a natural compound, what we can do is that we can take an enzyme that generates histamine called histidine decarboxylase and express it in a presynaptic cell, and then co-express the transporter protein that can transport histamine into synaptic vesicles. And we're doing this in a cell that normally does not make or use histamine. And so we're co-expressing with the endogenous neurotransmitter. And so then the idea is that when this cell normally gets stimulated and releases its own neurotransmitter in red, it'll then co-release histamine, all right? And then on the postsynaptic side, what we can do is that we have the normal endogenous receptor, but then we can co-express the transgenic histamine receptor, okay? And so if this is a histamine-gated chloride channel on the postsynaptic side, and then the normal endogenous transmitter binds to an excitatory receptor, this neuron gets excited by the normal receptor, but then the histamine blocks that excitation by inhibiting. And so then you lose the signal. And so you're essentially silencing the connection between these two cells. Okay. And so I decided to do this using this ASH and AVA connection here. And ASH is a sensory neuron that detects glycerol, and AVA is required for doing reversals. And so, I took the VMAT, the transporter from Drosophila, and expressed it in ASH. I also took an enzyme from Drosophila that makes histamine and co-expressed that there. And then in the postsynaptic cell AVA, expressed HisCL1. The idea is that when this gets activated, normally you'll get a reversal, but then because we're co-releasing histamine onto the cell that has HisCL, that will then block the response, okay? And so ASH histamine by itself has no effect on reversal length. Everything is normal. HisCL by itself in AVA, no response, and everything is normal. Put them together, you should see the response go away if it's working. Sadly, it does not work. Okay, and so that was unfortunate. And so then we did some more thinking about what might be going on. And so what it turns out in Drosophila, HisCL actually forms a partnership with the HisCL2. And so there's actually a heterodimer that's the endogenous receptor. And when we express the endogenous heterochannel in worms, they're actually tenfold more sensitive to histamine than just the HisCL1. And so I thought, let's just use the endogenous heteromer. Let's just use the heterochannel and see whether that works. And so when I co-express HisCL1 and HisCL2 in AVA, now the system works, which is very exciting. And so what that suggests, the initial failure was because we weren't producing enough histamine, because the Drosophila system was evolved to work with this much more sensitive heteromer, okay, with both HisCL1 and 2. The problem is that when we co-express HisCL1 and 2 in worms, that heteromer all by itself has effects. There's some leaky opening. And so this is where we express it pan-neuronally in all the neurons simultaneously. HisCL1 on its own has no problems whatsoever. It's even without histamine, there's no effect. but HISCL1 and 2, if you co-express it, the animals are slower. And so we don't want to use the HISCL1 heterochannel in the worms. And so what I did is that I needed to find a way to improve histamine production and release. And so to do that, we used a different assay, which was developed with my colleague, Steve Flavell. And so if you put an animal on a lawn of bacterial food, she will then crawl around this lawn. And this is called, it's called roaming. And then you can count and quantify how the area that she roamed. Okay, it's a very simple assay. And this is dependent on a neuropeptide called PDF1, which acts through a neuropeptide receptor, PDFR1. And this is a G protein coupled receptor that activates G-alpha-S, which then increases the production of cyclic AMP. Okay, and then that stimulates roaming. And so if you delete either the peptide or the receptor, these animals dwell. They don't roam. They just sort of sit in one place. But we can then replace the PDFR receptor with HRH2, which is a mammalian histamine receptor that activates G-alpha-S and causes an increase in cyclic AMP. And so if we express this receptor and then put these worms on media that contains histamine, these animals now roam. Okay, and so we were able to substitute for the endogenous receptor with the histamine receptor. And the reason this was useful for finding a better way of producing histamine is because the PDF peptide is produced far away from the cells it actually signals to. So you get extrasynaptic transmission. And so the PDF producing cells are not directly synaptic. They're acting humorally. And so we can express the receptor and add exogenous histamine to do the roaming. But if we want to find a system that produces a lot of histamine, what we did is that we did a screen where I took VMATs from a number of different organisms and co-expressed them with histidine decarboxylase enzymes from a number of different animals as well as bacteria. And then I screened for all these different combinations using these assays. And I found a couple of combinations that seemed to work well. And even without adding histamine exogenously, it caused roaming. And so here I have cells that have animals that produce histamine in the PDF in the PDF peptide cells. They don't roam. If I express the receptor, they don't roam. But if I then cross them together and put them into the same animal, where now I have cells producing histamine, and then I have cells that respond to histamine, now they're able to roam. And so, and this is again a extrasynaptic signaling. And so that means we're producing enough histamine to go beyond the synapse and go everywhere. And so it suggests that it works. And so then what I did is that then I transplanted the optimal VMAT and this bacterial enzyme into ASH, the glycerol sensory cell. So this is histamine version 2. And then when I express HISCL1, which is just the single channel, not the heterochannel in ABA, now the system works even with just HISCL1. And so that suggests that now we've succeeded in producing a better system than what we had before. But one sort of monkey is that the system might be working too well. And so what I show in black here is the normal non-transgenic animals. And what you can see is that the histaminergic system actually roams more than the normal system. And so we actually get hyper roaming. So it's actually, we have too much of a good thing now. And so we need, so now this is a challenge, is how do we deal with that? And this also seems to be the case with this individual connection. And so this AVA cell is required for responding to touch, as I discussed before, and to the glycerol. And so if you take normal animals and you give them, and you touch them, they'll do a reversal. And then if you can then give them a drop of glycerol, they will do a reversal. And then if you wait 1 minute after you give them glycerol and you touch them, they'll actually decrease their response slightly, because the idea is that they've run away from glycerol, which is a danger, and they're going away. And now you touch them again, they're sort of like, well, I just ran away from something bad. Maybe I should just sort of ignore this for a while and just keep going. And so you get a modest decrease, but then 5 minutes later, the touch response is completely back to normal. But in my synthetic histaminergic system, when I touch them without glycerol, there's a decreased response already. Then when I give them glycerol, that response basically, and then I touch them again, that glycerol response goes away. And then I have to wait like 15 minutes for to basically come back to a normal response. And so what we think might be happening is that normally what happens is that with normal neurotransmitters, the neurotransmitters are cleared. So they're released, and then they're cleared away from the synapse, okay? And what's happening here is that we're releasing this histamine, which is then binding to the postsynaptic HISCL or the postsynaptic HRH2 in the roaming assay. But the worm, it doesn't have any mechanism to clear away the histamine, just like animals that are evolved to use histamine as a neurotransmitter, okay? And in fact, this is kind of the opposite problem of what people that might have depression. And so in a lot of these individuals, it's thought that they have a low level of serotonin. And so there are drugs, these SSRIs, that block this reuptake, artificially then increasing the serotonin level. And so what we think is happening is that our animals, these artificial animals, have excess already. And so we would like to then go back to here where we get some either reuptake or degradation of the histamine, okay? And so what we're doing now is we're screening through, we've cloned a bunch of these histamine degradation enzymes, and now we're screening through to find ones that now reduce the response to something that's more, make it more like a natural neurotransmitter. And so I'd like to thank Taesuk and the others for inviting me. And again, I want to thank the folks that were involved in helping me, both from Corey's lab and then also the students at New York Tech. Thank you very much, and I'd be happy to discuss and take questions.
Fantastic talk, Navin. I love especially the videos, which I believe are related to the topic of Nobel Prize in Physiology of Medicine, receptors for touch and other signals, although not histamine, right?
Right.
So any question from audience? I believe, Nathan has question. You could unmute.
Okay.
Or I could ask the question for you.
Yeah, no, I'm happy to ask the questions. 1 second. So yeah, Navin, I thought that was super interesting talk.
Thank you.
So one of the things that you pointed out was just that the strength of synapse, really didn't predict, how much something was going to have an influence on a behavior. And at one level, that does kind of make sense, right? And it's, it might not be a property of a particular interaction or a particular node or something like that, but it might in fact be a property of a system.
Mm-hmm.
Just kind of curious, if you've looked at things like network motifs or, predictive algorithms, and partly what I'm thinking about here is a lot of the translation of health from the, like from worms into human. people often want to take like a molecule or a observation, and like you're pointing out, like that very often may not translate, but there's probably a level of, understanding the circuitry that does. So I just kind of love your thoughts about that, given how much work you've done in this space.
Sure. So and there's been a number of works using, like some of the earliest network analysis from the late 90s used the wiring diagram of C. elegans. And so there's an overrepresentation of these feed-forward motifs, where you have A goes to B, A goes to C, and B goes to C. So there's an overrepresentation of that in the worm wiring diagram. And also turns out there's also an overrepresentation of those sorts of connections in artificial circuitry used in electronics. And so, but until we have the ability to, actually manipulate individual connections, we don't have any way of testing that, testing the functional importance of that. And that's actually something that I'm very excited about investigating using using this technology, is to actually ask, like, what are these feed-forward motifs?
Yeah,
in the context of a nerve system. And so with that ASH neuron, it's sort of at least in neuroscience, right? And there's a lot of work done on connectomics in humans and in mice and fly. There's in the Nature this week, there's this massive like consortium paper where they're looking at the connections from a motor cortex in the mouse to basically all over the brain. They do single cell RNA-seq and see like where all of these go. And so there's a massive amount of effort and resources being spent on getting connectomic information. But I sort of the modeling that I just showed and what your expectations is that, like just because you have a lot of connections that or strong connection that might not be predictive. And what I'm hoping is that by cutting individual connections, we might be able to see like what are better correlates than just synapse size or synapse number. But right now, I don't know. I mean, that's the short answer. And even with the simulated network, right? I don't know. And certainly with the biological one, I don't know yet. But I think having both the simulated network and the biological network, hopefully together, we might get some clues on that in the next few years. And that's the great hope. I do. You also, so one thing might be important are the roles of these hubs. And so, and this is true in genetic networks as well. You have some neurons are some genes or some neurons or some proteins are hubs where they create a lot of connections. And so you might have some neuron that has connections to a lot of cells, and it might have a lot of connections to one particular cell. But the one that's really important is the few or tenuous connections it has to a hub cell. And so then if you lose the connection to the hub, then you're in trouble. And then what these other connections are might be more modulatory. And so they might be important in certain contexts. Does the
connectome topology, does it have kind of this characteristic topology we see all the time in nature of power law on the outputs?
Yep, absolutely.
On the inputs and
absolutely. Yep. Scale free and just like all these other
the usual stuff. Yeah.
All the usual stuff. And the worm was based. The worm wiring was one of the first examples of that in a biological system.
Right.
Yeah.
So,
are there? Yeah, I'll defer to let other people ask questions. I could keep going.
Yeah, actually, I also have some question, but I just checking the time right now is past the 5, and then I just want to check any other question from audience before we close. I'll ask a question to you later on because we have another Zoom meeting soon. So I just checking chat. There is no question. Okay, so so in that case, I'll ask some question later on. I mean, the another Zoom meeting. I mean, and then I need to probably close because it's already 4 minutes after 11 my time. So thanks all for joining and staying today again. And next week we will have Dr. Stephanie McHenry, the program manager, Army Center for Synthetic Biology, and Maria Eugenia Inda from MIT Team Lab. And see you next week in the same day and time. And the follow-up Zoom chat will occur via the link in the chat box.
So I should go to the chat. I should click on that Zoom link now.
Yeah. So you will click that one, and then you will just automatically move to another Zoom, and then you will just basically I will meet you there. Just with just second, and then great. If you wanted to continue discussion, you are welcome to join us. And also if you have another commitment, you could go.
I'd like to, but I actually have to jump to my next thing here.
Right. I mean, you're a busy man, so So I mean, so see you just one second later, and then also Nathan, see you next week.
Nice to meet you, Nathan.
Nice to meet you too. We'll have to.
We'll have to maybe meet
New York here.
Yeah, thank you so much. I'll visit you, or both of you probably in New York because you are both conveniently located similar. Actually, I'm going AICHA. I'm going to the DC and for my 10-day or 11-day trip. I mean soon.
Oh wow!
Right,
that sounds great. Yeah.
Yeah. All right, I'm going to jump over to the other session then.
Sure. So just click that one, and then I'll reach you there. And then some people also will joining there. Okay.
Okay.
Great. We'll see you then.
Thank you. Thank you so much, both of you.